A lot of great interactions with James as well. Thank you for inviting me to come here and tell you about the research that's been going on in my lab for the past 4 years. I forgot to switch it on, so thank you. You're like motioning to me and I'm not getting it. Just like, I'm gonna go ahead, I'm gonna keep talking. So I wanted to thank the organizers for giving me a chance to come here and tell you about what I work on. So what you can see here is, no, that's wrong. Okay, wrong button. That's fine. What you can see here is our maize epidermal cells. And these cells, oops, okay, I'm not gonna press that button either. I'm working on it, okay. You can see them dividing, okay. And we use kind of quantitative live cell imaging to understand how plant cell division contributes to growth and development. Now it's gonna play, that's fine. Okay, so one of the major topics that I'm going to be telling you about today is division plane orientation. So I wanna get you in the mindset by giving you a few brief definitions about what I'm talking about. So symmetric, I'm gonna press the correct button here, I swear. Okay, that's a good button. Okay. I got it, sort of. It doesn't look like it, but I got it. Okay, so division plane orientation. So what happens during division plane orientation, this is when the plant cell divides. It's going to divide typically symmetric, so 90% of the, or maybe even more of the divisions that occur in plants are so-called symmetric or proliferative divisions. So these lead to cells that have the same fate, right? So they just make more cells. These are symmetric divisions. And they play a critical role in growth, in plant growth. So you can have divisions that occur along this axis. This is a transverse division. And this is a longitudinal division. Well, I'll tell you more about that later on. But I can't forget to mention one of the key critical types of division that occurs in plant cells and of course across both bacteria, archaea, and also the eukaryotes, which are these asymmetric divisions. So asymmetric divisions are typically, asymmetric, that's nice. They don't have to be actually physically asymmetric, but they lead to changes in cell fate, and these are critical for plant development. But what you don't see very often are examples like this, where the daughter cells are equivalent size, but indeed you have a long plane of division. So typically, plant cells will minimize their division plane. And I'm gonna show you more data on that soon. Okay, the reason we're studying division plane orientation is because, as I was saying, it's critical. I'm not sure what's gonna play here or not. So I'm not gonna worry about it. The buttons, okay. Division plane orientation is critical for plant development, as I was talking about, but also bacterial development as well as animal cell development. So I want to give you just one case where division plane orientation could give us some insight into metastasis in human cancers. So metastasis causes about 90% of cancer deaths. But the very first steps that lead to metastasis in humans are not that well understood. One of the proposed mechanisms for metastasis, which is when you have a cell that escapes from its original tumor and goes somewhere else, one of these proposed mechanisms is aberrant division plane orientation. So typically you have cells that divide along this plane here so that both of the daughter cells remain fixed to the substrate, so they can't move anywhere. But if you have misorientation in this division, suddenly you have cells that are no longer bound to their substrate, and they're free to proliferate and then also to move. So by understanding division plane orientation, we might get some insight into this very earliest stage in human cancers. Okay, but primarily when we think of a division, we think of a constrictive mechanism of division. So typically, so this cell on the, I got this. Okay, this, come on, show my movies, please. Okay, Here we go. Okay, so here you can watch a C. elegans cell undergoing division. And what you hope you can notice, this is on the left-hand side here, what you can see is that the division occurred by constriction to separate the two daughter cells. Now plants, on the other hand, have a very unique method of dividing their cells. And this is really cool. What you have is the formation of a new cell wall that's formed inside the middle of a cell. And then this new cell wall, as it's being created, let's see if I can get the movie to work. Ta-da! You'll see the formation of the new cell wall here, but then it has to reach the correct edges, right? It has to reach the correct site. And we'll call this site the division site. So how does it do that? Okay. I did move forward, but now you can all see it. So to really get into this talk, I'm going to need to give you a couple of terms that you're going to need to remember. There will be a quiz at the end. Okay. No one laughed though, right? Everyone's nervous about this. But the two keywords that I want you to remember are the pre-prophase band here. So the pre-prophase band forms before prophase, pre-prophase band. It forms before the onset of mitosis, and it's a ring of microtubules and microfilaments that encircles at the very edge of the cell, but encircles the nucleus. So this pre-prophase band is thought of as kind of a hub or an organizing center for a lot of different proteins that accumulate here. But then, mysteriously enough, it's disassembled. It's disassembled right before metaphase to help form the spindle here. So in anaphase, you have the chromosomes separate. This is just a review, just to remind you. But in telophase, you have another plant-specific structure that's called the phragmoplast. So the phragmoplast is composed of two anti-parallel arrays of microtubules that kind of promote vesicle movement to form this new cell wall here in the middle of the cell. So the interesting thing here is that the phragmoplast unerringly returns back to the division site, which was originally specified by the pre-prophase band. Okay, so the pre-prophase band is pretty important, but then it's disassembled. Okay, that doesn't make any sense. But luckily, there are a handful of proteins that have been identified that even though the pre-prophase band disassembles, are maintained at the division site throughout the entire process. We think that these proteins play a critical role in recruiting this structure, this phragmoplast cell plate structure, back to the correct division site. So we're very interested in understanding more about these proteins. Ta-da! Okay, so I wonder if this is going to loop. Maybe not. It's a little bit sad. Okay, I'm going to go back again and try it again. So what you can see here is a maize epidermal cell expressing a live cell marker for microtubules. The arrows are here to indicate the pre-prophase band. Everyone's getting an A so far. Okay, and what you can see is that it goes away, and then you have the formation of the new cell wall in the same place. So you might be wondering, I don't see a band here. What's going on? Well, if you bisect the middle of the cell here, these two bright dots are actually the pre-prophase band. That's the pre-prophase band. That's what you want to look at. Okay, I think we've seen that enough. We know that the pre-prophase band is critical because mutants exist where the cells never make a pre-prophase band. And you can see, I'm not going to belabor this point because it's pretty hard to see in this. Well, that's fine. Okay, what you can see, though you barely see, is that the patterning of the root is totally altered, okay? So this is a wild type Arabidopsis root right here, and this is a root missing protein that promotes formation of the pre-prophase band. So these cells still complete cytokinesis. They're still perfectly able to divide their cells, but the patterning is altered because this pre-prophase band is missing, and the pre-prophase band is critical for giving information to the phragmoplast about where it needs to go. So to understand more about division plane orientation, we use mutants. So one of the mutants that we have in the lab is called a tangled mutant. So does anyone tell me what's wrong with this mutant, just from a large-scale viewpoint?
It's all jacked up.
Yes, exactly. But in what way? But in what way?
Short.
It's short, right? Okay, so this would be, we would talk about this in our ontology, right? We talk about the phenotype. Ontology is short. It's got a short stature. And if we look using scanning electron microscopy, what we can see is that the patterning of the cells are misoriented, okay? So the patterning is misoriented, and we think that this is due to a defect in division plane orientation. I mean, in fact, we know that, so I don't know why I said that. Okay, so what is tangled? What is the protein itself? So tangled is a plant protein that has kind of weak similarity to this C-terminal end of this human tumor suppressor called Adenomapolyposis coli. You can tell why I shortened it to APC. The interesting thing here is that APC mutants also have defects in division plane orientation. So from mouse stem cells or from Drosophila, these APC mutants also have defects in division plane positioning. So maybe we can get some insight into this enormous protein. This thing is over 2,000 amino acids by looking at something that's a little bit smaller and more tractable. Right. So I showed you a picture before showing that tangled localizes to the division site. This is one of those positive markers of the plant division site. And this is what this looks like in real life. So this is a maize epidermal cell with a live cell marker for microtubules. So you can see the pre-prophase band here and here. You guys can see that, right? This guy. Right? For all of you on this side, I feel really bad, okay? Okay, but what you can also see, what I hope you can recognize is that, that the tangled protein also localizes to the division site during metaphase, and it's maintained at the division site during telophase when the phragmoplast is expanding outwards towards the cortex. So we think that it serves this really important role in promoting proper division plane orientation. So we could come up with two different hypotheses about why the tangled mutant has defective division plane specification. So in one scenario, either it doesn't make pre-prophase bands, that's one option. You saw that mutant already. That's not the case here. So, but either maybe it forms the pre-prophase band in the wrong spot, like this. If it forms it, can you guys see that? Gosh, okay, there's no point, okay. So, sorry guys, okay. If you form the pre-prophase band in the wrong location, then you'll have new cell wall formation also in the wrong location. So what am I talking about wrong location here? This pre-prophase band doesn't minimize this surface here. It doesn't predict a minimal division. So that's what I mean when I say aberrant pre-prophase band formation. Still separates them into two equal daughter cells, but that's not enough. Okay. Now, alternately, you could have a defect after you've already formed the pre-prophase band, and this would be just that the phragmoplast doesn't return back to the division site that was specified by the pre-prophase band. So I'm going to give you a hint right now. There will be no quiz on this because I'll show you the data in a minute or a little while. That the primary defect of the tangled mutant is actually that its phragmoplast doesn't return back to the division site specified by the pre-prophase band. But when we started looking at tangled mutant cells, and this should come as no surprise because this is what you saw with the scanning electron microscopy as well, that when we outline the cell walls, in purple here, and we look for the pre-prophase bands, you can see immediately that some of the tangled mutant cell shapes are very aberrant, right? They don't have a kind of rectangular prismatic shape that you would expect of typical maize epidermal cells at this stage. So the question is the pre-prophase band actually in the correct location? And what I mean by that is that it will form a minimal surface that will separate these into two equal daughter cells. So the way that we're kind of looking at this really, really, it was a weak method. Here's the angle. Okay, the angle between
I think it's missing. I don't know.
long cell axis versus the location of the preprophase band. So you can see in cells that have aberrant shape, this is going to
yes, you can. Yeah, you're such a good person.
preprophase bands that look classically as if they were misoriented. But that didn't seem very satisfying here, because I think there's at least a chance based on these images that these daughter cells will be separated into 2 equal volumes. We just needed to find out. So we took a mathematical modeling approach, and this was in collaboration with Ken Brack, who's a professor of mathematics at University of Susquehanna University in Pennsylvania. Okay, so we used, you've heard this, you know what I'm gonna say, okay? The daughter cells will have an equal volume. These are the rules. I got this. How can I have trouble halfway through the talk? Okay, that's fine. The daughter cells will have 2 equal volumes, and then also that the division plane that's formed, and I shouldn't say axis, because axis is 2D. We are so over 2D. We're in 3D right now, okay? The division plane will follow the shortest local path. So this was actually a hypothesis established by this guy named Herrera. Look at the date on that. 1888. Herrera is the first person who hypothesized, and what he said specifically was that plant cell divisions mimic soap films. Okay, soap film minimization is how plant cells divide. Okay, so this is a hypothesis that's been around for a long time. We decided to actually test this hypothesis using real 3-dimensional plant cells. A lot of the other modeling that's been done has used 2-dimensional plant cells. But we all know, everyone sitting here knows why people use 2D imaging, right? Because it's easy, but it's not very accurate, right? Yeah, I see some people nodding, you know what I'm talking about. Okay, so we used a soap film minimization approach. So let me show you. So this is actually a soap film. Isn't this cool? Take a look at that. So, but it turns out that if you have a plant cell that's shaped like this, the divisions that it will undergo are here, here, and here. And when we look on a population level, we can see that that's true. So it seems like soap films are a good way to predict plant cell division. And they've also been used to predict divisions in retinal cells, for example, in Drosophila. Where am I going with this? Okay, right, so how did we do this? Oh, you can't see it, that's a surprise. Okay, we took images. This sounds really trivial. I'll make it sound like that for now. We extracted the 3D volume of the cell. This was not as easy as we would like. We also extracted the location of the preprophase band. Why? Because this is where the plant cell thinks it's going to divide. That's the wrong term, but you know what I mean. So then what we did is, and this is in collaboration with Ken Brack, so we used his program, which is called Surface Evolver. It's freely downloadable from the internet from, and Julie, this is for you. This program was developed in 1992, okay? Ken is a mathematician, so he's using it to look at very strange cell shapes. So I think when he saw our cell shapes, he was like, oh, okay, cool, cool, cool. What? But so what we do first is we kind of smooth the surface to be able to define this volume using a formula. That's critical for getting this to work. We want to describe the shape as a formula. And then using a process that's known as gradient descent or soap film minimization, we can predict the division. What we do after that, of course, is compare how well the preprophase band lines up to the predicted division. So let me show you what, oh, it works, okay, great, good news. Okay, so this is soap film minimization in progress, okay? What we do is we cut the cell at 241 different planes so that we can equally sample the entire volume of the cell, and then perform iterative soap film minimization on all of them. okay? So what's gonna happen is, regardless of where you start out, you're gonna end up with just a few types of divisions. This was very satisfying from our perspective because these are the divisions that we see in vivo. These are the divisions that we see plant cells doing. They divide in a transverse orientation, and this would be like the global minimum for a rectangular prismatic cell, right? You can see that here. They can divide along a longitudinal axis. This is a local minimum along a different plane. And occasionally we can see that they form periclinal divisions as well. Now, if you're a plant biologist, specifically if you're like a developmental biologist, you get very excited when you hear the word periclinal division, right? Andrea, am I right? Yeah. Because these periclinal divisions are strongly correlated with new developmental transitions. Okay, so we were kind of excited to see all of the different types of divisions that we could see. So I'm going to make what was a long and very painful story kind of short here so that I can move on to some other things. But what we showed recently is that this soap film minimization can accurately predict a lot of divisions. And when it couldn't accurately predict divisions, in some cases we could see immediately when we went back to the original micrograph why. So it turns out that plant cells will divide according to a soap film minimization very often. But if there's an adjacent cell here, you can see this, there's a adjacent cell wall. I have to point this out. Dang, okay. Yeah, you can see that right there? Mm-hmm. Oh, you guys are all awake. Thank you so much. Okay, it's after lunch. I'm sorry. It's great. So anyway, so what you can see here is that, that the pre-prophase band lines up very well with the predicted division when there's no four-way junction that's going to be formed. So a four-way junction is when you have an adjacent cell wall here. So what ends up happening is that the pre-prophase band itself shifts away from a four-way junction. So typically you would have the division occurring here, but it doesn't. So this is to increase structural stability. This is just an idea, a very old idea, right? About why plant cells don't form these four-way junctions to promote structural stability, just as you would if you were building a brick wall, right? You don't have your bricks lined up like this, right? It's gonna shear. Okay, same thing is true with plant cells. So we could demonstrate that the pre-prophase band is offset in this case, but we were really interested in looking at other divisions as well. So we collaborated with lots of people on this. We did this one in-house. So we looked at ligule cells. So thank you for introducing that, Andrea. That's great. So during ligule development, there's a stage at which the cells divide periclinally. We were really interested in seeing whether or not the division that occurred in these periclinal divisions was predicted by a soap film minimization or not. This result was a little bit of a surprise. So what happens here is that, so this is a maize ligule cell, and then this is, if you're looking into the surface of the tissue, you can see that there's a periclinal preprophase band here. Okay, this is actually really hard to segment, so this was really fun. No, just kidding. But what you can see here is that it's actually the cell shape itself that promotes this periclinal division. So this is interesting because it means that the expansion is the thing that drives the alteration of cell fate, promoting this global minimum division along a very unexpected axis. We also looked at Arabidopsis guard cells. So this is in collaboration with Charlie Anderson's group at Penn State. These also divide according to soap film minimization. And indeed, because we're curious to see whether this was, whether we could use any cell, literally any cell, to look at, we also collaborated with Bob Goldstein's group. He studies C. elegans embryo development. In symmetrically dividing C. elegans embryo cells, you can see that we can get good soap film minimization for these cells also, and these are the global minima. Okay, so It's kind of a kind of universal-ish, universal mechanism of dividing a cell. It does, well, it's not really a mechanism though. That's one thing I want to make really clear. It's not actually a mechanism. Okay, it's a model. It's a prediction, right? So let's cut back to the chase here. I'll remind you what the question was at the beginning before we took a little detour. It was a really fun detour, and we've got a lot of work to do on it now that we can see the power of this mathematical model. The big question was at the beginning though, do tangled mutants place their pre-prophase band in the right spot, yes or no? So you can see here that the cell shape is aberrant here, but the pre-prophase band lines up pretty well with the predicted division. Oh, and one thing I should have mentioned, which I didn't, so sorry I've been showing you a bunch of graphs where you don't know what's going on. Okay, that's fine. So what we do is we actually measure the distance between the pre-prophase band and the predicted cut. And the lower the number is, the more accurately they match up. Okay, so low numbers at 0 means they match perfectly. When they're higher than that, it means they don't match perfectly. So most of the time, tangled mutant cells place their pre-prophase band in the correct spot. But there is a difference, and we're working right now to figure out whether that's a difference in cell shape or what. We don't have an answer for that yet. Okay, so let's go back to this. This is just to remind you where we're at. So they have a little bit of a defect in placing their pre-prophase band, but the primary defect is actually in moving this phragmoplast back to the division site specified by the pre-prophase band. So I'm going to show you some movies, which are all running. Cool. Okay. I might have to play these over again. So what I want you to notice is that this division is a wild-type maize cell division. The pre-prophase band was here, and the new cell wall is here. In this tangled mutant cell, which is very aberrant shape, the pre-prophase band was here, and the new cell wall is here too, okay? And this happens about 60% of the time. But about 40% of the time, instead of, I really have to show this movie here, because, you know, it's going to make people happy. Okay. Maybe. It's really quick. Did you see that there? You saw it. Hold on. Let me try one more time. Okay. So there's the pre-prophase band right there. Right here. Boom. Right? So the phragmoplast actually goes to the wrong site, and this leads to this really big defect in division plane specification. So indeed what's happening is that the phragmoplast doesn't track back to the division site originally specified by that pre-prophase band. Okay, so while we were doing all of this imaging, what we noticed, I'll show you the data. Yeah, no one wants to see data right now. Come on, show me another movie. But what we noticed when we were doing our time-lapse imaging was a kind of unexpected delay of the tangled mutant, both in metaphase. So wild-type cells take about 40 minutes to progress through metaphase. So you can see that here. This is a histogram showing the average amount of time that, or not the average, but the actual data, the amount of time it takes for these cells to progress through metaphase. So what you can see here is that wild-type cells take about 40 minutes, but the tangled mutants take quite a lot longer to progress through metaphase. Okay, we don't have enough time resolution in our time lapse to tell whether there's a significant delay in anaphase, but you can also notice that tangled mutants also take significantly longer to progress through telophase. This is caused by two separate things. One is that the phragmoplast doesn't return to the minimal division, so obviously it takes longer, it's taking a longer path. But also the rate of expansion of that phragmoplast is also slower, okay? So why is that? So I've shown you that tangled localizes to the division site, but that's only half of the story here. So it turns out that tangled also co-localizes with the metaphase spindle. So this is the spindle here. This guy? You guys all know what a spindle looks like. Yeah, totally. And then you can see a weak accumulation of tangled in the spindle. And you can also see a weak accumulation of tangled accumulating here in the phragmoplast. Okay. So it looks like tangled is located in 2 different locations, one of them which is at the division site, but one is also in the mitotic microtubule arrays. Okay, so what we did is we knew that tangled was a microtubule binding protein, but the way that it was shown was kind of non-canonical. So what we did is we in vitro labeled, and I'm not showing you all the quantification, but you'll see it soon, hopefully. Okay, we have in vitro labeled microtubules here. So this is totally in vitro, just a few different components, just microtubules and just tangled protein, both of which are in vitro. So if we reconstitute microtubules in vitro and we add tangled protein that's fluorescently labeled, you can see that it accumulates along the microtubules. So this indicates that it directly binds microtubules. And the really interesting thing is when we add tangled to dynamic microtubules, so these are growing in vitro again, they're growing and falling back as microtubules do. They grow and they collapse. What we can see, I wonder if these movies are gonna play right away. Of course they are. Okay, good. This is good. What I want you to look at here is cross-linking, okay? When we add tangled to microtubules, we see this unexpected unexpected behavior, which is cross-linking or bundling of microtubules. So you saw that those were 2. Let me see if I can play that again right quick. Okay. Oh, wow, check it out. Ta-da. Ooh, okay. Right, with the sound effects, so you can really see it. Okay. But it also has this other really interesting feature, which is that I hope you can see here that even as the plus end is retracting, that it can actually pull on another microtubule. Okay, so this indicates that tangled kind of cross-links or is able to interact with microtubules. And now this is a great collaboration that we have with Ram Dixit, who's out at Washington University, to answer these questions. So our current hypothesis about what does all of this mean, this in vitro microtubule binding, what does it mean, right? What does it mean for the mutant phenotype? What we think is that, that we know that there's a delay in mitosis. We know that tangled accumulates at microtubules. So the idea is that the reason for the mitotic progression delay is because tangled is missing, so it can't cross-link or have these microtubules interact with each other at that stage. Okay. So, of course, the next question that we were interested in asking is, okay, is there any correlation between mitotic progression delay and division plane specification? That would be really tidy, right? We could say, we would know already by metaphase that there's going to be a problem, so that the phragmoplast would end up going to the wrong location. But when we asked whether or not that was the case, so we correlated metaphase time with eventual aberrant division specification. What you can see is that there's no correlation. So either in metaphase nor in telophase when the phragmoplast is expanding. So we have this hypothesis that division plane defects in the tangled mutant are really due to tangled function in phragmoplast guidance, not due to microtubule dynamics delays. So the mitotic progression is because of delays in, mitotic progression delays are due to altered microtubule dynamics, but division plane specification is not. So one of the ways that we tried to address this was a molecular trick to remove tangled exactly at this stage of the cell cycle during anaphase. Okay, so the idea is we have tangled, it co-localizes with the pre-prophase band, that's nice, but then if we can eliminate it during anaphase, then we'll be able to see what role it plays during telophase when the phragmoplast is expanding. This is when we see our problem. the idea is, of course, that then we would see misoriented phragmoplast, and then this would phenocopy. This would look the same as the tangled mutant. That was the idea. So how do we do it? We fused a destruction box from the cyclin B. Okay, this is bringing you all back to very peaceful times. Okay, but the cyclin destruction box mediates degradation specifically during anaphase. So if I had a control, I constructed both of these. The control, of course, has mutations that don't allow degradation during anaphase. So this movie, you're not going to be able to see it because that's the break. But what you can see here is that this destruction box, when it's fused to tan-YFP, that it still localizes to the division site as we expect, and that over time, this signal goes away. Okay? The question is, when does it go away? Right? We would hypothesize that it's going away in anaphase. That's when it's being targeted. But it turns out it takes a lot longer for this protein to be destroyed. So we wanted to ask whether it could rescue the mutant phenotype. So first, I just want to remind you, these are maize cells. They look pretty normal, okay? And then there's the tangled mutant. It looks pretty abnormal. When we introduce the tan-YFP transgene, you can see that really good rescue. This is in the tangled mutant background. You can see it localizes to the division site. That's nice. Sorry. The control does the same thing, also rescues. To our surprise and general horror, okay, this is terrible. Okay, the tan destruction box fused to YFP also rescued, and it rescued across multiple different transgenic events. So we were like, why is this happening? But we did see one, and we were excited about it. We were excited about this one, lucky number 13. Okay, destruction box, tan-YFP, event number 13, because it looked like it had a division plane defect. So we thought, oh, aha, here's the one. We can use this one to understand tangled's function during telophase, because obviously it has misoriented divisions. This must be the problem with it. But when really talented graduate student Pablo Martinez did 106 time-lapse images, this took a long time, don't ask. It turns out that only one of these divisions was actually misoriented. So this mutant, even though, or this transgenic rescue line, even though we were excited about it, had a very moderate defect in division plane orientation when we quantified it. Why am I telling you this? This is like a sad story. And it's not. It's not a sad story. This is a very interesting story, because what this particular event did is it allowed us to mostly uncouple mitotic progression defects from division plane specification. It wasn't what we were hoping to get, but it sure is interesting in the end. Okay, now I'm going to show you that, so if we look at the fluorescence intensity of the tan-YFP protein, you can see it here. This is where we, oh, okay. The glare on that's not good. Sorry guys. Okay. Oh, now I can't see it here. Okay, maybe it's just me. Okay, when we measure fluorescence intensity here at the division site, when we measure that for tan-YFP, you can see that it has this kind of, this is totally arbitrary units, but that it is maintained at the division site throughout the entire time. Now, lucky number 13 had low expression at all stages of the cell cycle, but especially low expression during telophase when the phragmoplast was expanding. So we thought, okay, maybe this is really telling us about a telophase specific role. Okay, but we found out that wasn't the case, and I'm going to show you the data proving that right now. Okay, right now. Dun, dun. Okay. So it turns out that if you expect something to function during anaphase or telophase, you can look earlier on in the cell cycle. We already knew that the tangled mutants had delayed progression through metaphase. Okay, so it took them about 61 minutes on average. Lucky number 13, when it's in the tangled 1 mutant background, takes an intermediate amount of time between wild type, which is about 40 minutes, and then this is about 50 minutes, okay? We also did that for telophase. Okay, it also has an intermediate delay there. So, but then we wanted to look at where this protein localized, okay? Okay, so here I just want to remind you for a second that tangled accumulates in the metaphase spindle and also in the phragmoplast. It accumulates in the mitotic microtubule structures, okay? But the destruction box fused to tan-YFP localizes only to the division site, or it's below detection at the metaphase spindle and also at the phragmoplast during telophase. So, of course, this our simple-minded hypothesis here was that, of course, this means that we've lost kind of this hazy low accumulation just because there isn't enough protein, right? There just isn't enough protein to go around. So it's targeted to the division site, but there isn't enough of it to go to the mitotic arrays. So our hypothesis was, okay, so we have this mutant that has a 1.3 slower mitotic progression delay, very minor defects in division plane orientation. We thought naively, okay, this plant is going to be intermediate in size between the tangled mutant. Remember, that's a short one, right? Short, that's the geo term, right? And between the super tall wild type plant, we thought it would have an intermediate growth. And it turns out that wasn't the case at all. So we measured, this is just one leaf, but our reviewers were pretty particular. So we measured lots of things. More things than I want to think about. But in all of those cases, what we saw was that the destruction box tan-YFP, lucky number 13, when it was either in the wild type background or in the tangled mutant background, that it grew just as well as wild type plants, just as well. Okay, there's the tangled mutant there. It's pretty obvious, a little bit smaller, right? Okay, so what this really meant was that this mitotic progression delay doesn't really have a big effect on the plants. Why? Because plants are great at compensating for mitotic progression delays. So what that tells us, this is the last thing. I know I've packed your head full of lots of stuff. So here are my conclusions. The first conclusion that I want to make is that we can uncouple the mitotic progression delay from the phragmoplast guidance defect. We could show that those plants grow just as well as wild type plants. So this suggests that the combination of a mitotic progression delay and a division plane defect is actually critical for growth. Now, you guys work on really phenotypes that have massive importance for agronomic traits. For me, this is very exciting to talk about how something that I think of as a very fundamental process, division plane orientation, might actually be essential for growth, whether it's directly or indirectly, we don't know. And then I talked about these other things. We know that soap film minimization can be used to predict divisions, and I showed you that tangled can cross-link microtubules in vitro. But anyway, let me wrap it up and just say thanks to the people who did the work. A vast majority of the work that I talked about today was done by a really talented graduate student, Pablo Martinez, together with postdoc Hong Liang, technician Lindy Alsman, and 2 undergrads, one of whom was a REU undergrad in 2015, so he's now a co-author on the paper. So get your students started early. And also thanks especially to the NSF for funding it. And with that, I'll take your questions.
All right, do you have any questions?
It was so clear.
So I've got one for you.
Okay.
So it'd be last week, our journal club actually reviewed your latest review that's coming out here in 2019. And so we were checking it out and we were kind of like, you know, a lot of us are very applied, very like in the field doing stuff.
Yeah.
And so we're trying to like ballpark, what could we do with this idea, this concept? So we're curious, do you think this could be applicable if we could locate these genes could we maybe start to look at leaf architectures where we could localize these planes? Is that something that this might be applied to in the future?
I think that I'm glad you read the most recent one, which is on typically focused on asymmetric divisions. I think so. What I told you before was that like 90% or maybe more than 90% of the divisions that occur in a plant are these proliferative divisions, right? But I think in order to have a big impact on important crop traits, it might be more useful, just like as Andrea was, as Dr. Evelyn was saying earlier, that it might be more useful to push certain asymmetric divisions instead of to work on the kind of core proliferative division machinery. Because there you can promote specific, kind of more specific gene expression, cell fate patterning, which is going to lead to, of course, more or earlier. I don't want to say anything too specific, right? Because you know, but it could lead to better yield, for example, at the end. That's a nice vague way of saying it. But by partitioning, really by partitioning the energy use that the plant has towards something that we find useful, right? So.
Yeah. Interesting. Thank you.
Sure.
Do we have any other questions?
Yeah.
Oh, okay. Is the process of cell division different in the main stem versus the leaves? Because the leaves looked fine. It's just the stem was just kind of chopped off.
So I wish I could show you the leaves in person, because the leaves are also totally messed up. And every single place that we looked where there are dividing cells, we see misoriented divisions. So we really think of tangled, at least, as one of these nuts and bolts division plane machinery proteins. So yeah, the leaves are also the altered and smaller, and they take longer to develop. But eventually, you know, they do, right? They form kind of morphologically normal leaves. So this suggests that the plant has a plan, right? The plant has a plan, and even though there are local defects in division plane specification, it can still accomplish a lot of its overall architecture goal.
So we can't just make a short cord.
Wouldn't that be great? That would be great. But these plants, I will also say that these plants are a little, they're not very tough, right? I wouldn't want to, when we put them in the field, they're not quite as sturdy. They're not quite as sturdy as wild type maize. So I'd hesitate to, I see where you're going with this, and I love it. And you know, maybe we could think about controlling the expression, but I think maybe it would be better to go after, you know, genes that are important for expansion, for example, first. Yeah. Yeah.
You can slow down the cell division, and you can speed it up.
It's always easier to break something, right, than to make it faster. I think we can, I think there are a lot of clever tricks that plant biologists have done to do exactly what you're talking about, to either not, to increase the number of cells, for example, by hijacking some of the cell cycle machinery, for example. That's a pretty easy way to do it. But whether you can speed up mitosis, anaphase, and telophase, all I can say is just make it warmer. Right? I mean, if you want to talk about a really trivial way to do that, but from a genetic standpoint, I'm not aware of mutants like that. Yeah, that's a great question.
Awesome. Do we have one last